you.
Welcome back to the Early Career Immunology Seminar Series. For those of you returning, thank you for your continued support. The purpose of this seminar series is to increase the exposure of early career faculty in a time which we had not yet achieved equity and representation at scientific conferences and seminar series, despite equally innovative discoveries. If you sit on an organizing committee for conferences or seminar series, consider selecting some of our speakers from our lineup from the past year. We're excited to have Bella Rauch with us today. Bella did her PhD in the Koffler Lab at the University of Salzburg in Austria, and there she developed extensive expertise in the antimicrobial activity of certain neuropeptides produced by keratinocytes in the skin. Bella's postdoctoral work was then carried out with Thomas Decker and then subsequently with Russell Vance, and focused on understanding inflammation and pathogen restriction in the intestine. Bella joined the faculty of the Department of Molecular Microbiology and Immunology at Oregon Health and Science University in Portland in 2019. And here her lab focuses on animal models and stem cell-derived organoids to interrogate epithelial cell biology from various angles, because every cell is an immune cell. If you have any questions for Bella, please type this into the chat box and Justin will address these at the end of her talk. Bella, thank you for being here with us today.
Hi, thanks for that introduction, Tim, and thanks very much for Tim and Justin to organize this series, and for the whole organizing group to invite me to give a talk here. I think it's a really great platform for us new-ish professors to talk about our science. And today I want to tell you a little bit about what we've done in the past, and then at the very end I'm going to talk about some very recent stuff in the lab that deals with a very specific subset of intestinal epithelial cells. So, as most of you are probably aware, tissue barriers, and especially the intestinal barrier, face a huge challenge in our everyday life, because our intestines are covered by this one layer of intestinal epithelial cells that on the one hand is supposed to take up nutrients and water and other things, but on the other hand, of course, needs to keep all kinds of microorganisms out of our body. And that doesn't only involve harmful pathogens, but of course also our regular harmless or even helpful microbiota. And of course, the intestinal epithelium has evolved a couple of things that help it with that job over the millennia. There is a nice mucus layer covering epithelial cells that keeps a lot of microorganisms that are not really motile out of it. Antibodies are being secreted in the lumen that sometimes block the more motile bacteria. And of course, there is antimicrobial substances that are being secreted to especially protect the stem cells that are down here in the crypts of the intestinal epithelium from anyone getting near them. However, the problem with professional gastrointestinal pathogens is that these also had some time to evolve, and they are very often resistant to antimicrobial factors. Many of them are mobile, so they're pretty good at burrowing into this mucus layer. And most of them have some kind of secretion system that allows them to inject effectors into the cells that they infect. And that often allows them to either manipulate the cell so they can adhere to its outside, or even actively invade the cell. And many gastrointestinal bacterial pathogens have also evolved a bunch of effectors that can manipulate the immune signaling that happens after a pathogen has been recognized. So just passively keeping them out is definitely not an option, but the immune system also needs ways of recognizing gastrointestinal pathogens. And if we just look at the overall strategies that the innate immune system uses to recognize pathogens, the one and probably most famous one is the detection of microbe-associated molecules, so the famous PAMPs, which, like for example, toll-like receptors, just recognize features of microbes that are not present in our mammalian bodies, and then that leads to an immune response. However, the problem with this strategy is, in general, that it can, of course, also be activated by non-pathogens because like, for example, LPS is obviously also on harmless members of our microbiota. So this is where the second strategy comes in, and that is the detection of pathogen-encoded activities. So this is a more specific because it recognizes a behavior of a pathogen that usually leads to some kind of damage of the host. And that is very specific to pathogens because by definition, commensals don't really strive to invade or damage the host. And one class of molecules that uses this strategy of pathogen detection is the inflammasomes. So just to give everyone a quick overview, inflammasomes are this whole class of cytosolic receptors that are in the cytosol in an off state. And when some kind of pathogenic activity is being recognized, they are turned on. And then this leads to activation of caspase-1. And caspase-1 is this protease that cleaves a bunch of substrates, importantly, the cytokines IL-1 and IL-18. And then the other one that's very important is gasdermin D, which when cleaved leads to pore formation in the cell, which then leads to pyroptosis, which is an inflammatory and lytic type of cell death that also lets these inflammatory cytokines leave the cell. And one inflammasome that we are researching extensively in the lab and that we're using as a model for inflammasome roles in general is the NAIP NLRC4 inflammasome. So this inflammasome recognizes bacterial products, which you could argue are PAMPs. However, it recognizes them in the cytosol of a cell, which makes them clearly a danger-associated pattern because they're in the cytosol. And it recognizes needle and rod proteins from these bacterial secretion systems, so type 3 secretion systems, via these NAIP proteins. And then there's another set of NAIP proteins in mice that recognizes bacterial flagellin. And humans actually only have one NAIP that interestingly recognizes all of these ligands. And then once the recognition happened, it leads to a structural change that allows oligomerization with NLRC4 and formation of an active inflammasome complex that leads to these downstream effects that I was talking about. One thing that I still need to introduce is a tool that we use extensively to study the inflammasome that we call FlaTox. So FlaTox is based on the anthrax lethal factor toxin, which is an AB toxin. So usually the way this works is you have the lethal factor protease that gets brought into the cell by the other part of the toxin, which is protective antigen through this N-terminal signal. But what we did is we replaced the whole C-terminal part of lethal factor with bacterial flagellin. So now this allows us to put it together with the protective antigen and this way deliver bacterial flagellin into the cytosol of a cell, which then leads to NAIP activation and NLRC4 inflammasome formation. And what's cool about this tool is it basically allows us to activate the NAIP NLRC4 inflammasome in isolation without activating any other innate sensing pathways, which would obviously happen during an actual bacterial infection. So it allows us to study the inflammasome in a very targeted fashion. And it was already shown before I got interested in using this tool to study cell type specific effects in barrier tissues that if you give this FlaTox systemically to mice, it leads to a very drastic and very fast sepsis-like phenotype that is due to systemic inflammasome activation, mostly in peritoneal macrophages. And that leads to the release of eicosanoids, which are these lipid inflammatory mediators from peritoneal macrophages, and rapid blood clotting, and that is very lethal. However, I was interested in seeing what the role of the inflammasome is in intestinal epithelial cells, because just by looking at the pattern of what this inflammasome actually recognizes, secretion systems and intracellular flagellin, it made it a very good candidate for epithelial cells to make this really important distinction between who's a harmless commensal that stays outside of me and who's a pathogen that's invading. So we made a mouse that allows us to study this inflammasome in a tissue-specific fashion. So this is the opposite of a conditional knockout mouse. It's a conditional knock-in that has a fluxed stop cassette in front of the NLRC4 gene. And then we cross this mouse that we call INLRC4 onto an NLRC4 deficient background. And now if we cross it to a Cree line of our choice, we can express NLRC4 only in epithelial cells or only in myeloid cells. So for epithelial cells, we cross it to William Cree, and then for myeloid cells, we cross it to the Lysem Cree line. And the nice thing about this whole system is that NLRC4 is completely dependent on the NAIP sensors. So we can't actually create any artifacts by expressing this in cell lines, because it only works in cells that already express the NAIPs anyway. So when we went and treated these mice with FlaTox, we found in our kind of positive control when where we crossed the mice to Lysem Cree, not very surprisingly, these behaved very much like wild type animals because, as I told you before, peritoneal macrophages have this very dominant phenotype when the inflammasome gets activated that basically kills an animal in half an hour. So if you look at the body temperature, this is 8 degrees of body temperatures that the animals lose in half an hour. However, interestingly, the animals also got really, really sick when we activated the inflammasome only in intestinal epithelial cells. And one way this manifested was also that the animals lost a lot of plasma. So this is an indirect readout. This is the hematocrit, so basically how thick the animal's blood is. And you can see that this goes significantly up, even if we only activate the inflammasome in intestinal epithelial cells. So the animals are losing a lot of fluid somewhere. And we also know where they lose the fluid, because if we look at the content of their intestines, so if we just take the feces of these mice and do a normal wet-dry ratio, so basically a very crude readout of diarrhea, we can confirm in numbers what you can actually see with your own eyes is that these intestines basically look like what you read in descriptions of a cholera patient. So they're basically just filled with fluid, and these mice get really drastic diarrhea. And because we knew that eicosanoids are associated with fluid loss, so these lipid mediators are incredibly fast acting because they're not transcribed and translated like proteins, but they're made in an enzymatic cascade. And so when we knew that when you activate the inflammasome in peritoneal macrophages, you get prostaglandin E2 release. So we analyzed the tissues of these mice by lipid ELISA, and we could actually see that there is this very significant release of prostaglandin E2 in the tissue, also when you activate the inflammasome only in intestinal epithelial cells. And then, of course, not very surprisingly for everyone that knows about inflammasomes, we also get really, really elevated levels of IL-18 when we activate the inflammasome in intestinal epithelial cells. So next, we wanted to see what actually happens to this in intestinal epithelial cells, because there was already a really cool paper by Mika Selin, who at the time was in Wolf-Dietrich Hart's lab, and had shown that upon Salmonella infection, you get loss of epithelial cells in an inflammasome-dependent manner. So we looked by histology at the intestines of these mice, and we saw this really, really drastic loss of epithelial cells. So you can see down here, the villi are still somewhat present, but they have shrunk massively compared to the NLRC4 deficient animals that are injected with FlaTox. And you can see all these intestinal epithelial cells that are being shed off the tissue. And because it was already reported that the inflammasome is necessary to protect from Salmonella infection this way, we wanted to see whether it's also sufficient to do this. So we used our knock-in model in a model of oral Salmonella typhimurium infection, where we look at tissue invasion. So this is the levels of Salmonella inside the intestinal tissue of these mice. And you can see that NLRC4 deficient mice here in white are very susceptible to tissue invasion. However, when we re-express the inflammasome only in intestinal epithelial cells, this basically brings the level of resistance to invasion back down to what it is in a wild type mouse. So the intestinal epithelial inflammasome is really the most important sensor in these early stages. So this is at 18 hours post-infection. However, if you look at the y-axis, you will also notice that there is still plenty of Salmonella that actually makes it through despite the inflammasome, because Salmonella is a very well-adapted pathogen that can actually invade mice and go systemically very well. However, two other members of the Vance lab, Patrick Mitchell, who was a postdoc there at the time, and Justin Roncaioli, who's a graduate student. And I have to highlight that Patrick has his own lab at the University of Washington now, and I think we should definitely invite him for the series and have him talk about his work. They got interested in what's going on with other gastrointestinal pathogens that are not known to infect mice. So it's been a huge trouble for the field that mice are more or less completely resistant to Shigella, which is this really nasty gastrointestinal pathogen that causes very, very bad bloody diarrhea in humans, and a very nasty disease. However, mice don't get sick from it at all. However, surprisingly, when they went and infected NLRC4 deficient animals and compared them to wild type mice, they did see a really strong increase in myeloid peroxidase in the intestinal tissue, which is a readout of inflammation because it's a major enzyme in neutrophils. And they also saw a really drastic weight loss in these animals in the first 2 days of infection. They saw that these animals get diarrhea. And they also, when they looked at colonization of intestinal epithelial cells, they saw a significant increase of colonization of intestinal epithelial cells. So now these animals actually reflect what we see in a human, and they actually get sick from Shigella if they don't have the NLRC4 inflammasome. And they then went back and asked the same question that we asked in our Salmonella model, which is the intestinal epithelial inflammasome sufficient for this? And indeed it is. So when they used these INLRC4 Wilkri mice again, so they were orange in the earlier data, they're red here now, they can basically rescue this intestinal inflammation measured by MPO back to baseline. And when they have a pathologist look at the inflammation in the intestine, this actually also significantly goes back down basically to baseline. So the epithelial NLRC4 inflammasome in mice is sufficient to prevent Shigellosis. So if we quickly recap what we've learned about the intestinal epithelial inflammasome so far, is we know that activation leads to cell sloughing and inflammatory mediator release. And that cell extrusion upon inflammasome activation, and I didn't have the time to tell you, is actually mediated not only by caspase-1, which is this classic effector of inflammasome activation, but I could show that it is actually partially dependent on also caspase-8, which we think is maybe kind of an evolutionary backup because this pathway is so important. And when we overly activate this pathway, it can lead to pathology. However, it does protect from Salmonella and Shigella invasion. However, a big question that we had was whether this is intestinal epithelial cell autonomous. So we know that these lipid inflammatory mediators are being released from the cells very fast. Is there some kind of crosstalk between the epithelial cells and the underlying immune system that leads to this extrusion? And does this actually lead to cell lysis? Because you would think it's somewhat dangerous to cause pyroptosis and a monolayer of single cells that is covering the intestine. So I went to do some experiments in intestinal organoid models. So these are stem cell derived organoids that we grow in a dish, and they grow into these nice, relatively lowly differentiated, the way we grow them, intestinal epithelial cells that are polarized. And then we can go and add propedium iodide as an indicator of cell lysis, and we can add FlaTox to activate the inflammasome, and then just look what happens. And if you focus on this arrow here, you'll see that there's a cell that gets permeabilized, and immediately after that, it gets extruded from the epithelium. So if we look at that in a still, you can see that this is actually a really fast process. So from the time that the cell gets permeabilized to the time it has completely left the epithelium, that's not even 5 minutes. And it is a very different process than when you look at apoptotic cell extrusion, which we can, for example, cause by treating with high doses of TNF, where the cells never lyse while they're still in the epithelium, but also get extruded. However, it kind of seems like this is also a much slower process than the pyroptotic cell extrusion. And Mika Salin's lab has actually shown really nicely in vitro models that there's this really cool contraction of the surrounding epithelium around pyroptotic cells that probably leads to a faster extrusion, which obviously makes sense because you really don't want a lysing cell in a monolayer for sitting around for a very long time. So we wanted to see whether this also happens in vivo, because of course organoids are great models, but you can always feel more comfortable with your model if you see it in vivo too. So we went and injected mice with FlaTox and then also with propedium iodide to look for lysing cells. And yes, we can find lysing cells in the epithelium. And if we zoom in on these cells and stain for actin, we can actually see this massive actin rearrangement around the lysing cell that is basically actively pushing out this dying cell of the epithelium. And to just formally show what every cell biologist is probably not surprised by, we repeated our organoid experiments with inhibitors of actin rearrangement. And you can see that now all the cells lyse eventually, however, none of them actually leaves the epithelium. So you really need this active actin rearrangement and this basically these epithelial cells working together to get rid of the infected epithelial cell. So this is the model that we have at this point, where we think that when an intestinal epithelial cell gets invaded by a gastrointestinal bacterial pathogen, it's being recognized by NAIP NLRC4, and then that leads to activation of caspase 1 and 8, and then that leads to this really rapid extrusion of the epithelial cells from the epithelial monolayer, and at the same time, you get release of these inflammatory mediators, IL-18, and then prostaglandin E2. So what I got really interested in is this question: is this actually a common response of all subtypes of epithelial cells? Because in our histology analysis and also in our organoids, which are really very lowly differentiated, we didn't really focus on any cell types other than the most common one in the small intestine, which is these regular absorptive enterocytes. But of course, there's a lot of specialized subtypes of epithelial cells that sometimes also have been reported to actually be the point of invasion for certain pathogens. And one piece of data got us interested in one very specific subtype, and that was when we looked at our intestinal tissue by lipid mass spectrometry. So I showed you very early on, we showed prostaglandin E2 release by lipid ELISA. However, apparently these ELISAs, especially if you use tissue samples, can sometimes be not super reliable. So our collaborators at UC Berkeley that know everything about lipids, the Gronert lab, offered to analyze our tissues by mass spectrometry. And very nicely, they could confirm that when we activate the inflammasome only in intestinal epithelial cells, we see this significant release of prostaglandin E2. However, what was really interesting is that we also saw this significant release of this prostaglandin D2. And that at first was very confusing because for this, I'm going to have to give you a little update on eicosanoids. So eicosanoids are these lipid inflammatory mediators that come in several flavors. Most importantly, the leukotrienes and the prostaglandins are two major groups. And they all start out made by arachidonic acid that is liberated. And then depending on which enzymes they get metabolized by, you get this end product of a certain eicosanoid. And in the course of making prostaglandin D2, what you need as a first step is either PTGS1 or 2, which you probably know more commonly by their names COX1 and 2. And then this intermediate product, PGH2, is further metabolized by specific enzymes. And to make PGD2, you need this enzyme HPGDS, which is commonly known as hematopoietic prostaglandin D synthase. So it was named this way because it was believed to be only expressed in hematopoietic cells. So it was kind of confusing that we would see this prostaglandin being made in our intestinal epithelial samples, where we are fairly certain that we only activate the inflammasome in epithelial cells. However, this is where Jakob von Moltke at the University of Washington came in, because he studies tuft cells, and he had the single cell, it's actually not single cell, I'm sorry, it's a regular RNA-seq data set where he separated tuft cells in the colon and in the small intestine from other regular intestinal epithelial cells, and looked at gene expression. And not only did he see that tuft cells express all the genes you need to form a functional inflammasome, so caspase, gasdermin, LRC4, and the NAIPs, but interestingly, they express really high levels of these eicosanoid synthesis enzymes, COX1, and especially HPGDS, so this hematopoietic prostaglandin D synthase. So in theory, tuft cells should be able to make PGD2. So what are tuft cells? There are this very specialized type of intestinal epithelial cells. They're named for this little tuft of microvilli that actually makes them stick out above other intestinal epithelial cells, as you can see in this cool picture. They're really important in control of parasite infection, as Jakob and several others have shown in the last couple of years. And really interestingly, they also undergo this massive amplification during a parasite infection. So this is, you can see how rare they are in a regular SPF mouse. But then when this mouse is infected with a parasite, you get this massive expansion of tuft cells in its intestine. They've also been shown in mice to be an important target of norovirus entry. However, their function in bacterial infection so far is completely unknown. So we had this idea that maybe tuft cells are have special functions upon inflammasome activation. And of course, one really important question we had in the beginning is, are tuft cells actually also showing this extrusion response? So we went back to our FlaTox injected mice and stained them with a marker for tuft cells, which is called DCLK1. And this marker stains tuft cells here in green. And now if you have a look, you can see that this tuft cell is also taking up propidium iodide, so it's lysing. And if you look at the actin, you can see we get this really nice actin rearrangement around it. And then in the DAPI, you can actually really see that the nucleus is also kind of already in the process of leaving the epithelium compared to other intestinal epithelial cells. So tuft cells are maybe special, but they're not special in this way. So they're not protected from extrusion of the from the epithelium. However, what about this PGD2 production? So for this, first we started with an in vitro model where we grow short-term cultures of intestinal epithelial cells and compare from tuft cell deficient and sufficient mice. And this experiment was actually done by Jakob's lab at UW. And we can see very nicely in this in vitro model that when we treat the epithelial cells with FlaTox or with ionomycin, which is just an inducer of calcium flux, which can also trigger eicosanoid release, we see this really nice release of prostaglandin D2 in the supernatant. However, when these monolayers don't have any tuft cells in them, the release of prostaglandin D2 upon inflammasome activation goes away. So this was a point where we decided it's worth looking at this in vivo, and this is where two great trainees in my lab, Renate Bauer from the who was a visiting student from the University of Salzburg, and Madeleine Churchill, who is a graduate student in my lab, came in to look at this in vivo. So they went and crossed our inflammasome expressing mice to the PAU2F3 Cre line, which is a Cre that is specific for Tuft cells. So PAU2F3 is a transcription factor that is specific for Tuft cells. This is a tamoxifen inducible Cre. And I already have to preface that this is a really hard experiment because, as I told you, Tuft cells are very rare in a regular mouse. This tamoxifen induction is definitely also not 100%. So we were actually super gratified that when we looked in the intestines of mice where we re-express the inflammasome only in Tuft cells and where we activate the inflammasome with FlaTox, we can actually see significantly increased levels of prostaglandin D2 in the tissue. And we don't see a significant increase of prostaglandin E2 just as a control. So it looks like this prostaglandin D2 release is really Tuft cell specific. And this data actually eventually made it in a manuscript together with Elia Teit-Voinus lab, because they had a really great story on the role of PGD2 in helminth infection. And I encourage you all to come back on May 10th to check out Elia's talk. I actually don't know whether she's going to talk about this story, but she has a lot of cool stuff going on, so it's going to be cool no matter what. But we are interested in bacterial infection and what the role of this Tuft cell prostaglandin D2 release could possibly be in a bacterial infection. So we wondered whether there is some kind of crosstalk of this lipid mediator with the immune system that helps against a bacterial infection. And there is precedence of lipid mediator crosstalk between Tuft cells and the immune system. So Jakob's lab actually has shown that Tuft cells can also make leukotrienes, so they can make LTC4, and they could show in a very cool story that this LTC4, when it's released from Tuft cells, signals onto ILC2s, and then that leads to IL-13 release, and that aids in suppression of helminth infections. So we were wondering whether the Tuft cell release prostaglandin D2 is also signaling to some type of laminar propria immune cell and the signaling can lead to an antibacterial response. So interestingly, if you look at single cell RNA-seq data from laminar propria that other labs have published, we could find in two different papers that ILC3s express the receptor for prostaglandin D2. So that, of course, would make a lot of sense. If I can give you a quick reminder of what innate lymphoid cells are and what they do, they come in different flavors, and we got different subtypes, and ILC2s are the ones that Jakob reported are being signaled on by LTC4, and those are actually really important in type 2 immunity and anti-parasitic infection defense. However, ILC3s are actually been reported to be really important in defense against bacterial infections. So one thing that they do is they release the cytokine IL-22, which then leads to, among other effects, the release of antimicrobial peptides by the intestinal epithelium. So we went into our model of Tuft cell specific NLRP4 expression and activated the inflammasome. And very nicely, we can actually see that activation of the inflammasome in Tuft cells leads to an increase of IL-22 in the surrounding intestinal tissue. And we wanted to be sure that this IL-22 actually also has a biological effect. So we also did some gene expression analysis. And very nicely, we can see that gene expression of these two of these signature antimicrobial peptides, Reg3 gamma and Reg3 beta, is significantly going up when we activate the inflammasome in tuft cells specifically. So now, of course, this model is still done with FlaTox, which is quite the sledgehammer, and some might make the argument it's maybe a little bit artificial. So we really also wanted to see what is going on during a regular bacterial infection. However, when we did first experiments with our usual Salmonella strain, which goes systemically very fast in mice and then gets mice relatively sick very fast, we really didn't see any differences in these early time points where we, if I remind you of what I showed you really early on with the all epithelial cell expressing inflammasome expressing mice, we actually see this really nice inhibition of invasion. But we figured that maybe this lipid release and communication with the underlying immune system is leading to a more long-term effect and not like an early defense mechanism like the cell extrusion, but it's more of a communication with the underlying immune system that basically leads to a response that then mops up bacteria that have made it through, because of course this extrusion response isn't perfect after all for most pathogens. So we used an attenuated Salmonella strain that gets cleared by wild type animals in like about 10 days. And when we look at early time points, so this is 18 hours after infection, we don't see a difference in invasion in the cecum tissue or in the ileum, so the lower end of the small intestine. However, when we go and take this out to 5 days, interestingly, we still don't see any differences in the cecum between LRC4 deficient mice and mice that only express the inflammasome in tuft cells, which is not super surprising because the cecum actually doesn't have very many tuft cells. However, in the ileum, where the tuft cells are most plenty, we do see an accelerated clearance of the pathogen. And actually, at this time point, nicely enough, about half the mice have already completely cleared, at least below the level of our detection. And then the other half also clearly has lower levels of pathogen in their tissue already. So we think that the signaling is leading to an accelerated clearance upon tuft cell recognition of bacteria. So just to sum up what I've just told you about, the NAIP LRC4 inflammasome in epithelial cells, when activated, leads to cell extrusion, IL-18, and eicosanoid release. So eicosanoids are these lipid inflammatory mediators that act really fast. Then intestinal epithelial cell extrusion reduces Salmonella invasion and renders mice more or less completely resistant to Shigella pathology. And tuft cells, interestingly, uniquely among intestinal epithelial cells, can produce prostaglandin D2 upon inflammasome activation. And the tuft cell inflammasome activation leads to an antibacterial response and an accelerated pathogen clearance. So now, if you're a little deeper in the field and paid a lot of attention, you might be saying, well, what is this response actually good for? So this is our current model. Of course, obviously, I haven't really shown you that prostaglandin D2 signals on ILC3s. So we're currently working on that. But we got pretty nice data showing that it leads to IL-22 release somehow. Because obviously all the other intestinal epithelial cells in a fully inflammasome sufficient animal would make lots of IL-18. And IL-18 has also been reported to lead to a very strong antimicrobial response. So why would the immune system need prostaglandin D2 signaling on top of that? Well, one thing is that many pathogens have actually been reported to suppress NF-kappa B signaling, which of course is a major downstream signaling pathway of IL-18. So one thought that we're having is that maybe the tuft cells activate this alternative circuit in case a pathogen knows how to block IL-18 signaling. And what's actually really interesting is if we look at another epithelial cell sequencing data set published by Haber et al., nicely, they confirm that Taft cells here at the top express really high levels of these eicosanoid synthesis enzymes. However, if you look here at the bottom where IL-18 is, it's really interesting that enterocytes, so the regular majority of intestinal epithelial cells, make boatloads of IL-18. However, Taft cells actually don't even seem to bother to make IL-18. So I think this actually kind of nicely supports this idea of this alternative signaling pathway. And then, of course, one other thing that I want to remind you of is that, as Jakob and others have reported, during a parasitic infection, you get a massive amplification of these Taft cells. And Elia's lab has shown that during an helminth infection, you actually also get other pathways of PGD2 signaling. So maybe during a what's evolutionarily probably not that uncommon, a co-infection with a helminth and a bacterial pathogen, this type of signaling actually becomes much more important because you just have many more Taft cells that can release these lipid mediators. Okay, so with this, I'm coming to my thank you slide. So I really want to thank my lab. Unfortunately, we just got some, fortunately, we got some new lab members, but we haven't taken a new lab picture yet. But I really want to thank Renate Bauer, who joined my lab for half a year for a visit during her PhD program and really got a lot of these very hard models going in the lab. And then Madeline Churchill, who is a graduate student in my lab and who really produced most of the data that I was showing you on the Taft cells. And then, of course, I want to thank our funders. And at UC Berkeley, all the data that I showed you in the first half of the talk was still produced in Russell Vance's lab, and they've also still been super supportive since I started my own lab. The Gronald lab is our great collaborator on all things lipid mediators. And at the University of Washington, I am collaborating with the von Moltke lab and Elia Tait-Voino's lab. And I really have to say, it's so great to have these guys as collaborators because they have been super welcoming into the Taft cell field, which was obviously new to me, and are really enthusiastic collaborators on all things intestinal immunology. And it's been really great. And now I hope I get some cool questions.
So we usually have a few second lag. We'll let them come in. But I think the fact that it was so well attended and so far no questions actually speaks volumes to how well that talk was. It was beautiful and clear. At least that's what Mark Jenkins always said, right? Oh, that must mean that it was absolutely perfect. So I'll just start with a theoretical question. So a colleague of mine recently published in the context of Salmonella infection that apoptotic cell death fuels those bacterial growth via specific metabolites and things that are released, various nutrients.
Yep. Remember that paper?
But the obvious question is, of course, Salmonella inducing other types of cell death, and it becomes sort of a complex ecosystem of interactions. And I'm wondering what your interpretation of that is and how pyroptosis might play in this context. Is it actually a beneficial sort of cell death process? It fuels their growth, or just big picture thinking.
Yeah.
Feel free to take that word.
I think especially when we talk about Salmonella, it's such an amazing pathogen, and it has evolved to take advantage of the immune response, right? It also benefits as like the Boimler lab and the Bündler's lab and others have shown has this really takes advantage of the inflammatory response because it's also resistant to a lot of antimicrobial peptides and likes to respirate whatever happens during the inflammation. And if you look at these immunohistochemistry images in the Cellin paper on Salmonella extrusion, you can actually see that the cells lyse, but they don't explode. And you can actually see Salmonella just like a little astronaut inside that cell being shot out of the epithelium. And it always kept me thinking that maybe it's just sitting there in its own little capsule and traveling down the intestine and just slowly the cell is dead, right? But it still gives it like a little protection. So in theory, if that cell gets flushed out all the way of the host and into the environment, it might actually give the bacteria something to live on for a while. So yeah, I wouldn't hold it against Salmonella to totally trigger this pathway on purpose, because it's clearly not entirely restricted by it. If you look at how many Salmonella still make it through in a mouse, so it's the response is much more effective against other pathogens that haven't evolved to completely like suppress this.
It's a beautiful concept too, I'll just say, because if we think about the amount of biomass that any sort of cell has relative to the size and biomass of a bacteria, the amount of nutrition that a single cell could provide is remarkable, right? I mean,
it's like you bring your own lunch package for a week.
Yeah. Oh yeah, I like that. All right, we got some questions. So Philip Starkle says, hi Bella, great talk and very exciting data. Would you expect that mice deficient in tough cells or with tough cell specific PGD2 synthesis, I think synthesis deficiency, not sure these exist, have impaired Salmonella resistance?
So we're currently breeding the prostaglandin D2 synthesis deficient mice. So we'll hopefully be able to answer that question soon. We haven't tested, and it's on our bucket list, the tough cell deficient mice with our pathogen clearance model. I know that people have tested it with the regular Salmonella and haven't seen anything, which kind of goes along with what we've seen. And I think it's not super surprising because, as I said, Salmonella is really good at circumventing a lot of these. So we're using this attenuated strain kind of as a model hypothetical, not as well adapted pathogen kind of. So we'll do both these experiments eventually.
So another question by Fishy Squid. They claim their name is Ed Meow, but I'm going to go ahead and say Fishy Squid. It's a fantastic name. Great talk. Question, Imogen is telling me that neutrophils also express the PGD2 receptor. Is that correct? And do you think there is an ILC3 first, then self-sustaining neutrophil, I think neutrophil response after, to clarify that?
I think that's totally possible. We haven't really looked on single cell gene expression in inflamed intestines. So I don't think directly in the lamina propria, there's that many neutrophils in a healthy animal. At least I hope. At least by MPO, we don't really get a signal. And it's definitely interesting that that receptor expression seems to be very tissue specific because, for example, in the lung, it's expressed on Th2 cells. Like Elia has some really cool data on its role during helminth infection and allergy in the lung. So it seems to be very tissue specific. So just because it depends where, I'm assuming that those Imogen neutrophils are blood neutrophils. So. But then neutrophils, I don't think, are known to change their expression very much when they emigrate into tissues. So yeah, maybe later when you actually get the inflammatory response, you could get totally different signaling cascades. And as I admitted, we haven't actually shown that PGD2 LC3 signaling just yet. It's in the works, but it's hard experiments to do.
So just to follow up on that, a colleague of mine here at MSK, Philip Niedheimer, who does zebrafish work, beautiful imaging on PGD2 work, is actually shown these beautiful near instantaneous responses of neutrophils migrating in response to these too. So I think that is kind of an interesting and apt question.
It acts kind of like a chemokine, basically. Yeah.
Yeah. And they come in, but then they reverse and go back. They don't actually, as a cell death person, both of us probably think of neutrophil death as their fate, but he kind of shows that actually they recircle back pretty quick. So anyway, I think that is an interesting point. So I don't see any other questions. Again, thank you, Bella. That was absolutely fantastic. And so this stream will be available immediately following. So if you wanted to go back and ask questions on the actual YouTube stream, please feel free to do that. We will try and circle back and check them and answer them as best we can. And again, apologize for the spamming, again, the joys of online talks. And thanks again for being here.
Thanks. This was great.
All right, we are